Hello all, welcome back to the BioRock channel. In this video today we will be learning about the other processes of fertilization. In the first series of the fertilization we have seen that the sperm travels and it undergoes capacitation where its glycoproteins are modified. Few of the glycoproteins have been removed along with the cholesterol and the semen plasma proteins, right? So what I'll do now is I'm going to draw the oocyte, okay? For instance, if this is going to be the oocyte, this will be our nucleus, which is going to be the secondary oocyte nuclei. Okay, so this is actually the secondary ovum, right? Now, so this is my secondary oocyte. So, yeah, this is our secondary oocyte membrane. Secondary oocyte membrane, and there are few of the lysosomes here. So these will be the lysosomes. Lysosomes, and they are having some of the hydrolytic enzymes. So I'll draw it as scissors. So these are the hydrolytic enzymes present in the lysosomes. These are the hydrolytic enzymes. Okay, and in this cell we can see even the smooth endoplasmic reticulum. So let me take the smooth endoplasmic reticulum. So this is here, smooth endoplasmic reticulum. Let me write it for you. Smooth endoplasmic reticulum, which is going to have the calcium ions here. Okay, children. So next what I'll draw is we will draw the glycoprotein, which is going to be the Zona Pelosita. Okay, so let me take Zona Pelosita now. So the next layer that I'm trying to draw is Zona Pelosita. So let me color the Zona Pelosita. So this is our glycoprotein layer. Okay. This is our Zona Pelosita having its receptors. Let me draw the receptors now. So these are the receptors here, which are being called as Zona Pelosita type 3 receptors are present on this. These are the species specific receptors. Okay, these are the Zona Pelosita type 3 receptors, which are species specific. Only the human sperms can only make a contact. Okay, so this is species specific, most important. You have to remember that no other sperms from the different uh, organisms can actually make a contact with these receptors or enter through the Zona Pelosita, because these receptors are species specific. They only accept the human sperms. Okay, so these are the Zona Pelosita ones, and out you can see the presence of the follicular cells, right? What are those follicular cells or the granulosa cells? These are the granulosa cells of corona radiata. Okay, these are the cells of the corona radiata. This is whole togetherly the corona radiata. So let me write this for you. These are the cells of corona radiata, the granulosa cells. Okay, children. And inside, what is this? I told you this is all the secondary oocyte, right? This whole togetherly is going to be the secondary oocyte cell, having all these hydrolytic enzymes, smooth endoplasmic reticulum, and having the nuclei, which is in the stage of what? Secondary ovum, right? This is going to be in the form of secondary ovum, the nucleus, where it is in a metaphase 2. Okay, once the sperm is going to That's it. I told you that the secondary metaphase is going to proceed and it completes its metaphase, that is meiosis second division, and it will be converted into definite ova. Right, children, these were being seen. And I even told you that here you're going to have some of the connective tissue which is holding these granulosa cells. And this is also going to have an acid which is being called as high hyaluronic acid, okay, in between this. Now you saw that there will be these sperms with the acrosomal end, with the nuclei, with the middle piece having lots of mitochondria and a tail swimming towards, and it is already capacitated. So it has a very good motility, right? It is approaching. So it easily makes a way through this, and now it gets fixed out over here. So let's fix our uh, sperm here. So your sperm comes in contact with or binds with this particular ZP3, that is Zona Pelosida type 3 receptor. Okay, so here it is binding with this. Okay, this is the first procedure. So what are you able to see? The first thing is the capacitated sperm binds with ZP3 receptors. Okay, so they are fusing with the ZP3 receptors here. And the next thing that you can see here is the moment they are going to, what you call is bind them, something happens here. That is, for example, let me take a page and show you. If this is going to be the membrane, okay, this is going to be the membrane of the sperm with the middle piece and the tail here. These two are going to fuse at some sites. The two membranes are going to fuse at some site. So it looks like this. So they are fusing at one side, fusing at one side. So it's going to be this way. Okay. They are fusing at one particular site. Okay. And moreover, what happens is there will be a lot of calcium influx inside the sperms now, due to which what happens is, if for instance, if this area is being in contact, this is in contact, all those areas which are in contact are going to be washed away completely. So now you can see that there are some apertures or pore like structures which are being formed here, right? It is just because of the calcium influx. Now what is happening is there are some of the enzymes that will be released from the sperm now. Okay, and these enzymes will be the acrosin, the hyaluronidases, these enzymes, as well as some types of proteases, which can actually digest the proteins, the membrane proteins. So due to the calcium influx into the sperm, now what is happening is these enzymes get released out, and all these enzymes are going to digest away the Zona Pellucida membrane. So that will be the magic that is going to be done. So let me show you how this is going to happen. So there is a fusion here that has happened, and you can see that the membrane is getting, what you call is, you can see that the membrane is going to get cut, and because of the which process, because of the calcium influx into it, and this has happened here. Because of this, there are some enzymes released like acrosin, which is going to digest now. See, now they are digesting. Oh my goodness, give me a moment please. So they are digesting this membrane. So see, it's been broken apart. So you can see that these are being broken. Who is that? The Zona Pellucida is getting burnt away. It's getting open. So there is a damage of digestion of Zona Pellucida. So it's digestion of Zona Pellucida now. This is going to happen just because of the enzymes like acrosin, the hyaluronidases, the proteases, and this reaction is being called as the first one is being called as capacitation, and the second is being called as an acrosomal reaction. This is the acrosomal reaction, children. And next, now what happens is, as the sperm and the oocyte membrane, now you know that the enzyme ZP3 also has got digested. Now what happens is your sperm is going to move ahead. Okay, so now your sperm is going to move ahead and it is trying to make the entry and make a contact with this particular what you call is oocyte membrane. So you remember that the thing here to remember is, you know already that the head portion has been perforated because of the calcium influx. So you're having the nucleus here, okay, and you're having the middle piece here, and this is a tail. So only the lower portion which is not digested, because you saw that it has got perforated, it has got lost its head, and is making contact with the posterior head membrane here. Okay, and now what it does is, the moment the sperm touches this particular oocyte membrane, right? This is a oocyte membrane here. So the moment it touches, what happens is, here the sodium ion channels will open. Okay, these will open. So when they are going to open, now what happens, there will be lots of sodium influx, right? So the sodium is entering into the cell, the sodium is opening into the cell. So as it moving into the cell, now what happens is, there is lots and lots of positivity inside, just because of this sodium. So there is positivity or positive, it's going to become more positive surface who the inside membrane of the oocyte. Okay, now the moment the sodium influx is being seen and they're increasing, now what happens is, the oocyte for the first time it is going to block. Okay, it is blocking the other sperms to attach oocyte membrane. That's it, it's been locked now. Oocyte membrane. And this technique is being called as a third procedure called as fast block to polyspermy. No more extra sperms can come and touch this. Okay, this is fast block to polyspermy. As once the sodium ions are being opened and sodium influxes the positive channel, the secondary oocyte is getting a signal saying that it has to now stop the entry of the sperms into the oocyte membrane to get fixed over there. Okay, this is most important to see that. So the first thing is, the acrosomal reaction is being triggered by whom? Triggered by your calcium influx, right? As well as you can even see that it's been triggered by your zona pellucida, right? And now you can see that the sodium, it has gone, it made a contact with the oocyte membrane and the channels opened out, the sodium is moving inside, it is becoming positive, it has blocked any more sperm to get into the oocyte membrane. Now later what happens is, this is going to actually, in the next stage you can see that the haploid nucleus will be released in. Okay, this will be the next procedure. So once the haploid nucleus, this is a male pronuclei, right? This is a male pronuclei has entered over here. The moment it enters, now what happens is, your smooth endoplasmic reticulum starts pouring calcium ions. It is starting pouring up the calcium ions. Now the calcium ions, once they are being poured out, what happens is, they are going to activate the lysosomes. So what is calcium doing? Calcium is activating the lysosomes. So now what happens is, all your lysosomes, now let me erase few of the things and show you what the magic is going to turn on over here. So what is happening is, first of all, your these, what are those? Mm, the calcium influx has happened. Calcium is activating the lysosomes here. Okay, they are activating the lysosomes here. And now what happens is, the lysosomes will go and fix to the membranes. They are going and fixing to the membranes. That is the next procedure that you can see. Okay, they're popping to the oocyte cortical membrane, this membrane, they're going to fix. So your lysosome has moved over here. Now what it does is, it is going to release out the enzymes, the hydrolytic enzymes are released out. Once the lysosome enzymes are being released out, what happens is these enzymes now they are going to degrade the whole zona pellucida along with the proteins, the ZP3 proteins and all, such that there will be no more what you call is innovation by these sperms now. Okay, so these enzymes have been released, and these enzymes, what are they doing now? They are degrading the zona pellucida receptors, proteins. And the second thing that they're going to do is they're going to harden the cell membrane, harden the oocyte membrane. This is going to be the magic. So no more polyspermy, and this has been called as slow block polyspermy, we are going to call. So now there will be no sperms entering in. Okay. And remember, children, the calcium is being released. The moment the calcium is released, one thing is it is activating the lysosomes, right? They are activating lysosomes. The second thing what they do is the moment calcium is being released, they are signaling the secondary oocyte. So the moment they get the signal, the secondary oocyte will complete its metaphase 2. It will complete the meiosis 2. Okay, it will complete the metaphase 2, meiosis 2. Now it will be converted into a definitive ovum. Definitive ovum. So this is also going to become a female haploid pronuclei. Already we have got the male pronuclei. Now these two they are going to fuse to form what? To form the zygote. Okay, children, this is a magic that is being done. So what have we learned here was first thing is the sperms have got capacitated. This was being seen by whom? By the mucus of the uterus, by the vagina, by the fallopian tube. Next, the capacitated ones, the capacitated sperm came, they binded with the ZP3 receptors. Later, what has happened? There was calcium influx into the sperms, right? There was calcium influx into the sperms, and later the sperm head has got perforated, later it has been broken, and later they release the enzymes like acrosin, hyaluronidases, proteases. They all have degraded and dissolved whom? They started dissolving the which what did they do now? They started dissolving or digesting the zona pellucida, right? And the next procedure that you could see was now the sperm moves towards the oocyte and it makes a contact with the oocyte membrane, and later the smooth endoplasmic reticulum pours up the calcium ions, right? And once the calcium ions are being formed, but before that, what is happening? The sperm this has not released the nuclei, right? So the sperm along with the oocyte membrane, when they have joined together, the sodium channels open, right? The sodium channels on the oocyte membrane are open. There will be sodium influx into the oocyte, right? And this will what you call is lead to the fast block to the polyspermy, and later the male pronuclei is being released into the oocyte, and later the smooth endoplasmic reticulum is releasing the calcium, and this release of the calcium is going to move the lysosomes to the membrane of the oocyte, and from there they release enzymes, and these enzymes are going to degrade further the zona pellucida 3 receptors and many membrane proteins, and they even harden the cell membrane. Okay, they harden the oocyte cell membrane, which we were calling as what? We were calling this as a slow, what is that? Slow block to polyspermy. And now what happens is the moment the calcium is being released, it is even signaling the oocyte nucleus to complete its to complete its meiosis too. And now later it will be converted into a female pronuclei. Okay, children, this is the way the female pronuclei has been there. So you have the female and the male pronuclei. This will result in the fusion and formation of a zygote. This is being called as fertilization. I hope, children, you all have understood this procedure. Let me see if I have some of these slides for you, such that I can show you and clear your concepts of the acrosomal reactions and the fast and slow block polyspermy. Okay, so let me see the acrosomal. Yeah, so acrosomal, you can see here. I have told you that you're going to have the binding with the ZP3 receptors, the calcium influx into the sperms, right? See, the calcium influx. And later these vesicles, the acrosomal like exocytosis, and this wall is going to be dissolved out. Now it will be moving further inside towards what? Towards the oocyte membrane. So this is acrosomal reaction. Next that I have is, I told you that during the acrosomal reaction, I told you that the two membranes they fuse and they form pits or perforated, due to which the head is going to be lost. I told you, so that is going to be the procedure here. The two membranes are going to fuse together, and at few points they are going to get perforated to release the acrosin, the high hyaluronidases enzymes to digest or degrade your zona pellucida membrane. And next thing is going to be the fusion of the membranes. As I told you, that the ions, the sodium channels, they open up when the sperm membrane and the egg membrane fuse together. I told you that the channels are being opened, and there will be more sodium rushing into the oocyte, making it more positive, right? So that is this fast block. Okay, that is the one. And I have these slides. Okay, so I hope you all have understood the procedure, children, of the fertilization. Please do hit the like button if you liked it, and try to comment in the comment section how you liked it. And please do share and subscribe my channel. Thank you all. Thank you.